Bismillahirrahmanirrahim. Students, today we have started our new chapter, that is inheritance. Inheritance means the transmission of characteristics from parents to offsprings. Parents pass characteristics to their young through gene transmission. Equal number of chromosomes from each parent are combined during fertilization. The chromosomes carry the units of inheritance called the genes. Now, what is genetics? Genetics is the branch of biology in which we study about inheritance, and it is the branch of medical science which is concerned with the transmission of characteristics from parents to offsprings. What are traits? These characteristics are called the traits. For example, in men, height, color of the eyes, intelligence are of inheritance traits. Chromosomes are called of a pair are known as homologous chromosome. Homologous chromosomes are apparently same. In human body cells, there are 23 pairs of homologous chromosomes for a total of 46 chromosomes. We may recall that during meiosis, the two members of each chromosome pair separate and each of them enters one gamete. Chromosome is made of chromatin material, simply known as chromatin. Chromatin is a complex material made of DNA and proteins, mainly histone protein. DNA wraps around histone proteins and form round structures called nucleosomes. DNA is present between nucleosomes. In this way, the nucleosomes and the DNA between them look like beads on a string. The fibers consisting of nucleosomes condense into compact forms and get the structure of chromosomes. Here you are watching the chemical composition of chromosome, and here is DNA, nucleosome, DNA between nucleosome and proteins which are histones. Now I will discuss about the Watson and Crick model of DNA. In 1953, James Watson and Francis Crick proposed a structure for DNA. According to the Watson-Crick model, a DNA molecule consists of two polynucleotide strands. These strands are coiled around each other in the form of a double helix, and there is a phosphate sugar backbone on the outside of this double helix, and the nitrogenous bases are on the inside. In this double helix, the nitrogenous bases of opposite nucleotides form pair through hydrogen bonds. This pairing is very specific. The nitrogenous bases adenine of one nucleotide form pairs with the thymine of opposing nucleotide, while cytosine forms pair with guanine. There are two hydrogen bonds between adenine and thymine, while there are three hydrogen bonds between cytosine and guanine. So here you are watching the Watson and Crick model of DNA and figure 15.2. There are polynucleotide strands which are double helix. There is a phosphate sugar backbone and hydrogen bonds. There is nitrogenous base, phosphate groups and sugar. And now I will discuss about how does DNA replicates. The DNA is replicated or duplicated It is done to make the copies of chromatids of chromosomes. During replication, the DNA double helix is unwound and the two strands are separated, much like the two sides of a zipper. So here you are watching, they are looking just like that of a zipper. Each strand acts as a template to produce another strand. Its nitrogenous bases make pair with the N bases of new nucleotides. In this way, both template strands make new polynucleotide strands in front of them. Each template and its new strand together then form a new DNA double helix identical to the original. So here you are watching, in first step double helix is unwound. These are the strands of parent DNA. Now strands are separated and new nucleotides are joining. Then there is a template strand. There are template strands and there are new strands at the end. Then there is a formation of new DNA. Next is, how does the DNA of chromosome work? DNA is the genetic material. It contains the instructions to direct all the functions in cells. It performs its role by giving instructions for the synthesis of specific proteins. Some proteins perform structural roles while the other act as enzymes to control biochemical reactions of cells. In this way, whatever a cell does, it actually controls its DNA. In other words, DNA makes the characteristics or trait of cell or organisms. So let us see how DNA is responsible for this. First step is, here you are watching, there are chromosomes, chromatids, centromere, a part of DNA, that is gene. In first step, the sequence of DNA nucleotides is copied in the form of messenger RNA. In second step, ribosome reads the messenger RNA sequence of nucleotides and joins specific amino acids. In third step, specific protein, that is structural protein or enzyme, make the characteristic of the cell and organism. So this is the working of DNA and it is also called the central dogma. Transcription, and this step is translation. The messenger RNA carries the sequence of its nucleotides to ribosome. The ribosome reads this sequence and joins specific amino acids according to it to form protein. So this is translation. The part of DNA, that is sequence of nucleotides that contain the instructions for the synthesis of particular protein, is known as a gene. So here you are watching gene, and DNA of each chromosome contains thousands of genes. Like chromosomes, genes also occur in pairs, one on each homologous chromosome. The location or positions of genes on chromosomes are known as loci. So here is, each gene determines a particular trait in an organism. Each individual carries at least one pair of genes for each trait. For convenience, pairs of genes are represented by a letter or symbol. So that was about the location of alleles on chromosomes. When these chromosomes are located on specific positions, then they will make, that is, albinism. It is a condition in which normal body pigments are absent. Like other traits, it is also controlled by one pair of genes. We can represent for these two alleles capital A and small a with capital A and both small a. These genotypes can be grouped into two types. The genotype in which the gene pair contains two identical alleles, capital A or small a with small a, is called homozygous genotype. The genotype in which the genes pair contains two different alleles is called the heterozygous genotypes. So here you are watching that a homozygous genotype is a cell is said to be homozygous for a particular gene when identical alleles of the gene are present on both homologous chromosomes. The cell or organism in question is called a homozygote. So a diploid organism is heterozygous at a gene locus when its cells contain two different alleles. So here you are watching that capital B is coming with small b. So there are two different alleles, one wild type allele and one mutant allele of a gene. The cell or organism is called a heterozygote. And homozygous you are watching that capital B is coming with capital B and small b is coming with small b, that are the alleles of these genes. Now I will discuss about dominant allele. When in the heterozygous condition, one allele masks or prevents the expression of the other, it is called dominant allele. The allele which is not expressed is known as recessive allele. So here on you are watching that capital B is coming with capital B, and capital B is coming with small b, so dominant allele here is brown, and the small b is coming with small b, so here the recessive allele is of gray. The effects of a dominant allele B are seen even if it is present with a contrasting recessive allele small b. The dominant alleles are represented by capital letters and recessive alleles by lowercase letters. Albinism is a recessive trait. It is produced when both alleles are recessive. In humans, allele capital A produces normal body pigments while the allele small a does not produce pigments. If genotype is capital A or capital A with a small a, the individual will produce pigments. On the other hand, if genotype is small a with small a, no pigments will be produced and the individual will be albino. In this example, you see that allele A dominates over allele capital A dominates over small a because it is the pigment producing allele, and the effect of small a is suppressed by capital A. The expression of this genotype in the form of trait in our example being albino or having normal pigmentation is known as phenotype. So phenotype is dealing with the characteristics of physical appearance. Genotype is the combination of alleles an organism inherits from its parents. Phenotype is the organism's appearance. So here you are watching genotype versus phenotype.